years. I love this article from the New York Times in 1908. This was Dr. Coley, who spent some time in the Lower East Side of New York. He was a surgeon who had a theory that injecting bacteria into tumors was a good idea. This was very bold in an era before antibiotics, but he did report some spontaneous remissions. I'm not sure how many actually did occur, but this did lay the groundwork for cancer immunotherapy, and so it's rooted in the very 19th century concept that we treat one disease with another. But his work was seminal, and it did lead eventually to new ways of treating cancer. This slide I find very sad. This was what melanoma looked like when Dr. Carvajal and myself started our fellowship back about 15 years ago. So to explain to you, this is a survival curve, and it's not something I like to show to patients, but melanoma used to be a very dismal disease, and you can see the proportion surviving over a period of 50 months is very low. And this sadly compared the two state-of-the-art therapies that were available at that time, two different chemotherapy regimens, one of them single-agent chemotherapy, DTIC, and something else that was called Dartmouth, which was combination chemotherapy. So this is what we were working with. Fortunately, that's no longer the case today, and so while melanoma remains a very serious disease, very difficult to treat, unfortunately we do lose many patients, nonetheless there is hope. And if you look at this chart, you can see the overall survival graph on the right that you do have about 50% of patients who are alive now at 2 years, whereas before it was less than 10%. The latest data with immunotherapy shows that at 5 years, about 35% of patients are still alive, which is quite a significant improvement over under 5%, which is what we had 15 years ago. So we really are making great strides in this disease, and we are so excited to move forward and to really, develop new therapies and improve those percentages even more. So what is the concept of immunotherapy? The concept of immunotherapy is really very appealing. It really harnesses the body's own defenses to treat cancer. The concept is that we all may have small tumors in our bodies, which very fortunately we are not aware of. And as those small tumors grow, they are sensed by the immune system, which in the first picture you can see the immune system in most cases is able to eliminate those cancers. In some cases, the immune system isn't successful in eliminating the cancer, but it is able to control it so that it remains microscopic and we never have to worry about it. Unfortunately, in a small minority of patients, the tumor cells do continue to grow and to escape the immune system. And really the purpose of immunotherapy is to reverse that process, to wake up the immune system that wasn't able to control the tumor, re-energize it, and allow the cancer to go into remission. So chemotherapy and immunotherapy have a very different mechanism of action. Chemotherapy is simply a toxin, and the purpose of giving it is really to directly kill the cancer cells, simply by poisoning the cancer cells. Of course, there are side effects associated with that. Immunotherapy is very different, and sometimes in some ways can be frustrating because it's an indirect mechanism of action. So the purpose of immunotherapy is to activate the immune cells in your body, and those immune cells then eject the cancer. Now, it can take a while to activate the immune cells. Sometimes it takes longer than chemotherapy, and so it leads to a little more unpredictability both for the patient and for the physician, but ultimately the results in melanoma are superior. One thing we do have to worry about with immunotherapy is that there's a reason why our immune systems aren't always turned on, and that's because if the immune system is too active, what happens is we get autoimmunity, and we can really get serious consequences of having the immune system being activated all the time. Arthritis, even diseases like lupus are caused by an overactive immune system. So the side effects we watch for with immunotherapies are very different from chemotherapy. We are looking for autoimmunity, and when we see autoimmunity, that can be a problem, and we have to stop the immunotherapy. So how does immunotherapy work? Well, most of our immunotherapies that are available today are designed to activate T cells. And you know that T cells are the cells that are attacked by the HIV virus. Now, T cells are very important immune cells in our body, and they have special molecules on their surface that can actually kill other cells. And in most of our lives, those T cells are important for killing bacteria or for killing other cells that are infected by viruses. But those T cells can also kill cancer cells. And so most of the immunotherapies we have available currently are designed to turn our T cells on. So, of course, most people who have cancer do have T cells that are capable of killing their cancer. And as we looked at in the earlier picture, this is part of the process of cancer development where at some point the T cells are no longer able to kill the cancer cell. But this slide is just to make the general point that there's lots of cell surface proteins and markers on the surface of T cells that can be manipulated, and some of these manipulations can effectively wake up the tumor cell. We don't wake up the tumor cell, wake up the T cell. And so this is why we've seen really a proliferation of therapeutics that are designed to activate all these molecules on the surface of T cells. So we have 3 antibodies FDA approved that activate T cells. These include one antibody directed against CTLA-4, which is ipilimumab, or the brand name is Yervoy, and also 2 antibodies against a molecule on the surface of T cells called PD-1. So those are just the names of the molecules on the surface of the T cells that are affected by these drugs. So first I'm going to tell you about anti-PD-1, and I'm starting here because this is really, at this point, I would say the most effective weapon we have in our arsenal against melanoma. So PD-1 is a molecule on the surface of T cells, and is really used by tumor, viruses, and normal tissues to quiet down the immune system. So this is a marker that's expressed on T cells when they've been active for too long, and the body decides, you know what, calm down, we don't have to worry about this threat, we are going to stop being inflamed. So this is taken advantage of by viruses. So in chronic viral infections, you will see T cells start to express PD-1, and it's also used by normal tissues to tell the immune system this is normal tissue and you shouldn't be attacking here, as well as, unfortunately, by tumors. So the strategy that this drug uses is it's an antibody, and it blocks that pathway so that the immune system is then active. So this is from one of our speakers today. This is from his paper, Dr. Hamid. But this shows that anti-PD-1 does help most patients with melanoma. This is a bit complicated, but I would focus on the panel A, and this shows you the change in the tumor burden for patients. And you can see that most patients treated with this drug will have decrease in the amount of tumor in their body. And some patients will, in fact, have elimination of the tumor from their body. Again, this is showing, that we're really seeing improvement in survival here. So this is an example of a patient who was treated with anti-PD-1. You can see that after 4 doses of treatment, this is a drug that is given every 3 weeks. You can see that the volume of tumor burden has diminished significantly. And this is something that we see very often, fortunately, with our patients who are treated with immunotherapy. It can take a while. So sometimes, the first scan might show shunt shrinkage, and then the next scan will show more and more. But we frequently see wonderful responses in our patients to this drug, and we're just so happy that we have it available. So what are the side effects of anti-PD-1? The side effects are pretty much as expected. We talked about autoimmunity, and so you can get any kind of itis from these drugs. And so we basically tell our patients, any symptom that's unusual, let us know, but the main side effects are colitis, which means inflammation of the bowel causing diarrhea, and we also watch carefully for any shortness of breath from inflammation of the lungs. And most people will have some fatigue on this medicine. So that tells you about anti-PD-1. I'm going to tell you about anti-CTLA-4, which was the first drug immunotherapy shown to prolong survival in melanoma, so very exciting. But it was the earlier version, and in many ways, anti-PD-1 is now something we turn to first, and we might turn to CTLA-4 if PD-1 doesn't work. So similar to anti-PD-1, the way this one works, it also blocks a mechanism that's meant to turn off the T cells. So what happens is that when a T cell is active and it's proliferating and it's attacking an infection, for example, eventually it will start upregulating on its surface CTLA-4, which is a control mechanism that's meant to shut it down. And animals that have deficiencies in CTLA-4 will have bad autoimmune disease. And in fact, it's been shown that some mutations in this gene are also associated in humans with autoimmune disease. But when we block it, what happens is the T cell will continue proliferating and dividing. And of course, in the case of cancer, that's really what we want. So this was a survival which was not as good as anti-PD-1, but this was really exciting for those of us in the fields when it came out a few years ago, because you can see that there's about 20%, there's a tail end of that curve there, where patients are still going on and living. So that was very exciting for us to see that everyone wasn't dying of this disease anymore, that there was a plateau there, and that then led to the anti-PD-1, which of course is much better than this. So CTLA-4 side effects are remarkably similar to PD-1 side effects, but they do tend to be a bit worse. And the reason for that is that the CTLA-4 mechanism is more fundamental to the T cell and not as specific to the tumor. So you can get side effects, and particularly we worry about the diarrhea and colitis with this medication. This is an example, a famous example now, I believe, but of a delayed response with immune therapy, in this case, CTLA-4. And this is to make the point that I mentioned earlier, which is that immunotherapy has a bit of a more complicated and indirect mechanism of action than does chemotherapy. You can see after the initial doses of treatment, the lesions in the liver actually got worse. And then, after the drug was held, they got better. And what happened there was, in fact, the lesions were becoming inflamed, because sometimes what happens is the immune system all of a sudden notices, wait, there's cancer here, and it sort of rushes in and causes inflammation, and that can artificially make lesions on scans look worse. I remember this patient very well, and I remember he told us that he was going on a camping trip, and he looked in great shape when he came in with that scan, which led us to suspect that the treatment was in fact working. So this can be a confusing phenomenon. I've seen it several times in my patients where the tumor actually looks worse before it gets better. But the key factor is that in all those cases, when the immunotherapy was working, the patient felt better. So the scan might look worse, but the patient would say, I'm feeling great. So that's an interesting thing that can happen with immunotherapy. Then, of course, why not combine A and B, right? So we definitely sometimes do give anti-PD-1 combined with anti-CTLA-4. It's not clear. It looks like it's a little bit better. It's not clear how much better it is yet. The studies are still ongoing to try to determine whether the combination, in fact, is better. But this is a dramatic example of a response. And you can see this horrible tumor that really just melted away with the combination immunotherapy. So sometimes you get very dramatic results with these immunotherapies. Then most recently, we have another new exciting treatment that was recently approved by the FDA, oncolytic virus. So this is a virus that is actually designed to kill tumor cells. So very exciting, and in a way takes us all the way back to Dr. Coley, right, and his idea that we treat one disease with another disease. So I'm not going to even say the name of this drug, but we call it TVAC. And TVAC is a modified form of herpes. So it was actually cultured from human cold sores, genetically manipulated to eliminate neurotropism, which means that, you know, one of the problems with herpes, of course, is that it can cause encephalitis, and we certainly don't want to be causing encephalitis. So it was genetically modified so that it does not infect neurons. And so it has been modified to make it more immune stimulatory and more likely to kill cancer cells. So it has two mechanisms of action. In a way, this is a bit similar to the radiation oncology talk we had, where the idea of the abscopal effect. But basically, it can directly kill the tumor cells because it's a virus, and it's able to do that. But what it also does is when it kills the tumor cells, it kills them, and a lot of herpes proteins are released at the same time. And then what this does is this works sort of like a vaccine, where it teaches the immune system, look, we've got this cancer, we've got the herpes, you know you're supposed to attack the herpes, so why don't you go ahead and attack the cancer while you're at it? And that can lead to regression of distant lesions that are not actually injected. And probably the underlying immune mechanisms for this, again, are similar to the abscopal effect of radiation. So this is an example of a response to TVAC, and this sort of illustrates the point, because obviously, unfortunately, this person had so much disease that we couldn't really inject every single lesion. But the point is that because of the effect whereby the vaccine effect, then the lesions, even lesions that were not injected, also fortunately disappeared. So, of course, now we're getting into the exciting area, and I think Dr. Abid is going to talk more about this, where we get to combine immunotherapies with each other. We already talked about combining CTLA-4 with PD-1. This is early results from a study combining the virus with CTLA-4. And you can see the results. This is, again, is showing, to explain this graph, it's showing whether patients' tumors get bigger or smaller. We can see that of the patients, unfortunately, 4 patients did have tumors that got bigger, but most of the patients did get smaller. So again, this is a much stronger effect than you would see with the CTLA-4 alone. And so, again, the idea and what we're really working towards at Columbia is working to combine treatments with each other so that we can really maximize benefit for our patients. Of course, as I mentioned at the beginning, about 35% of patients at this point as a field with the PD-1 were able to cure, but there's so many patients, sadly, we're not able to cure. And so that's one of the area of really active research where we're really looking in my laboratory and in many other laboratories to try to understand why do some patients respond and why do others not respond. So one area of interest is that we know that when tumors do respond, if you look in the pictures at the bottom, you can see in the middle panel there's a lot of immune cells that have come into the tumor. So one area that we're really interested in looking at is, well, how many immune cells are there before we start treatment? Because maybe if there already are immune cells there and we give treatment, then we have a head start and it's more likely to work. So one area that we're working on in our laboratory is in phenotyping the cells within the tumor. This is an example of a cancer, and the red marks the tumor cells. You can see there's CD3 is a T cell marker. You can see there's a patch of T cells in the tumor. And so we're working on studying tumors to figuring out spatially how are the immune cells living inside the tumor, where are they located, and we're really looking at our patients to see if this helps us to figure out why some patients respond and others don't. The other aspect is that there's, as Dr. Carvajal will be talking about with precision medicine, there are a lot of genetic aspects to this, and one interesting area is looking at the types of genes that are being expressed by immune cells within the tumor. So we extract the genetic material from the tumor and we use that to analyze the immune system using a method which is shown here, called NanoString, which is basically just a method to get RNA out of the tumor tissue. And so we did find that certain genetic patterns were associated with enhanced survival in melanoma. And this picture is really just to illustrate a concept, but the idea is that there are a whole network of genes, of immune genes within the tumor that may go up and down. We believe that the network shown in yellow might be particularly important, and we really want to get that particular network upregulated within tumors in order to see a response. So ultimately, I think in the future, everything is coming down to systems biology, right? To these sort of massive networks. And one area that's really interesting is looking at what type of changes do we need with gene expression within the immune system to see a response to tumors. And that's an area that we're really actively researching at Columbia. So I think I skipped it, but I just wanted to talk about what we're trying to do as a team. We're really working to personalize the treatment to get the best treatment we can for our patients. You know, best case scenario, we treat someone with a therapy containing anti-PD-1. So I recently, just very gratifying, I had a young patient who just, like gangbusters, responded to the anti-PD-1. That's all we had to do, and we continue that. But sometimes, you know, when the anti-PD-1 doesn't work, we try to mix it up with other therapies that are available, both conventional and immunotherapy. So I had a gentleman who unfortunately developed progression in the brain on anti-CTLA-4 combined with PD-1. We gave radiation, perhaps we got an abscopal effect, and now we're treating him with the viral therapy combined with PD-1, and he's responding. So we are really working hard to come up with combinations and sequencing in order to maintain disease control for our patients. And I really want to thank our whole melanoma team. Dr. Gaskin is an amazing dermatologist. I send my whole family to her. Dr. Carvajal, who's really a joy to work with. Dr. Gary Schwartz, Dr. Brett Tabak, who is our surgeon, and Vasile Horst, who is a dermatopathologist who I work with, as well as members of my lab. I'm really fortunate to have some wonderful students to work with. And I also just want to say a word of thanks to the patients. You know, it's just really a joy to work with many of my patients, and they have taught me many things as well, and I always carry that with me and to keep me motivated as you struggle to get funding for research and those kinds of things. So thank you very much for your time today.